movie we must all play. It's the unwritten law. This is the kinesin, which is sort of transporting cargo along a microtubule. And we have heard many talks on this, so I'll try to talk about a different aspect. So Anatoly and also Stefan and others have mentioned that, this is a cartoon. This is not the correct picture. In particular, there's a lot more stuff that's inside the cell. So in my case, I'll be considering cases where you have multiple motors attached to this cargo, teams of kinesins which walk towards the plus end of the microtubule, and then teams of dyneins which walk towards the minus end. They'll both be attached simultaneously, and we'll try to see what are the transport properties. So this movie should be playing. So these are just experiments on, so this is just to demonstrate bidirectional transport. These are in Drosophila S2 cells. These are Drosophila neurons. I think these are from Gelfand's lab. And you'll see that most cargo are moving in both directions. So there's both types of motors that are attached to the cargo, and then they do like this way and a certain time this way and a certain time that way. So bidirectional motion is ubiquitous. In spite of that, the cell manages to sort of achieve transport towards the direction that it finally wants the cargo to go. So there is directed motion in spite of the fact that you have both species of motors attached, and you see this sort of motion of the cargo. Okay. So this is just a cartoon. So okay, no, this is not a cartoon. These are structures of kinesins and cytoplasmic dynein. So they look very different, and in fact, as we have learned recently over the last 10 years or so, dynein also behaves very differently from kinesin. So this is the kinesin motor domain, which binds to the microtubule. This is the tail region, which sort of binds to your cargo. In contrast, this is the motor domain of cytoplasmic dynein. Here you have your microtubule binding domain, and then there is this sort of slightly floppy stalk, and then the cargo attaches on the other end. Okay. So I'll elaborate a little more as I go on as to how dynein is different from kinesins, and what are the implications for that for this sort of bidirectional motion. So this is my art project. This is my microtubule with a cargo. This is my cartoons of dyneins. This is my cartoons of kinesins, and this is the sort of picture that I'll try to look at. Okay, so as has been mentioned again previously in many talks, the prevailing picture that you have, or one of the major prevailing pictures that one has in order to explain bidirectional transport of this sort is this tug-of-war mechanism. So you have kinesins, teams of kinesins, which are trying to pull the cargo to the plus end. On the other hand, you have teams of dyneins, which are trying to pull the cargo to the minus end. They generate force on each other, and you get this tug-of-war. So this was done by Stefan and Lepowski and Melanie Mueller way back in 2005 or 2008, I guess. And then, so this makes one assumption that the force is sort of equally shared between all attached motors. We know biologically that is the case, and there were improved models by Professor Cross and Ambarish over here, which sort of asks about what happens if the force is not shared equally. And then there are consequences to that as well. For the purposes of this talk, I'll go on with assuming equal load sharing. So keep in mind again that this is an approximation. Once we do a better model, some of the conclusions will change, but we expect that the larger features that I want to tell you about will sort of stay the same. And so this theory has now been around for almost a decade now, and there has been a lot of experimental sort of corroboration that indeed tug-of-wars do take place in this sort of motor-driven transport. So this is experiments from Roop Mallik's lab, and here you have traces of the motor moving along the microtubule. So here is a cargo which was initially moving towards the plus end, then it gets into a sort of tug-of-war where it does not move too much. There's a slight negative directed slope, but not on the scales of this precessive motion. And then maybe it switches direction and starts moving towards the minus end. And in fact, they showed that during this tug-of-war stages, what you have is that your cargo itself sort of gets elongated, which gives further evidence for this hypothesis that you have these mechanical forces that are driving your tug-of-war. Again, these are similar traces. This is cargo carried by one kinesins. So you walk for a bit, then you stall, and then the motor detaches, then it attaches again, and so on. This is cargo carried by multiple dyneins. These are another set of experiments where you observe these are, I think, neuronal vesicles. So you have neuronal vesicles walking on microtubules. You can construct, they do it both in vitro as well as in vivo. And then you can construct chymographs of how the cargo is moving along the microtubule. So what they did was that they calculated the probability distribution of run lengths between reversal. So sort of between here to here, then again between here to here, and so on. And what they find is that you have there's a peak in the probability distribution, which sort of corresponds mostly to diffusive motion, which is, I think, they define as something of the order of 100 nanometers or so. You have cargo trajectories which are sort of stationary, so they don't move a lot. And then you have these really, towards the tail end of this distribution, you have these really long precessive runs. And if you see the distribution of these run lengths between reversals, you'll see that both in vitro as well as in vivo, the distribution looks fairly similar. The same sort of thing is probably going on. You can sort of ask how much of these runs fall into the stationary category, how much are diffusive, how many runs or what fraction of runs are diffusive, what fraction of runs are precessive, and so on. And then if you look at these precessive runs, you can ask how many of these runs are directed towards the plus end, which is on the top of this bar, and how many are directed towards the minus end. So it's something like 40% towards the plus end and 60% towards the minus end in these experiments. And again, these are fairly consistent across in vitro as well as in vivo experiments. So these are just some sample experiments. There's been lots of experiments that sort of corroborate this view, that you have a mechanical tug-of-war that is going on. You have teams of dyneins, you have teams of kinesins, they pull on each other, and then you get this sort of a tug-of-war. Okay, so is this the complete story? No, as again we have heard in many talks, there are essential features that this purely mechanistic tug-of-war model misses out. So Stefan talked about regulation. So you have various cofactors that affect how these dyneins and kinesins behave in vivo. And these cofactor proteins can drastically alter the behavior of kinesins and dyneins. We heard about some experimental talks on cofactors from Krishanu and Professor Gross. Then Anatoly talked about interactions between motors. So what happens if you have attractive interactions? What happens if you have repulsive interactions? And so on. Manoj talked a little bit about how dynein is a cooperative motor. So teams of dyneins behave cooperatively, whereas teams of kinesins behave non-cooperatively. At least that's what's seen in experiments. I'll talk about a slightly different way in which how often experiments can differ from this simple tug-of-war picture that we have in our mind. These again are experiments, I think, from Gelfand's lab in 2009. So again, these are Drosophila S2 cells. So here is a cartoon. So here is my normal cell. This tip ends are where the plus ends of the microtubules are. The cell body or the shaft ends is where the minus ends are. And in general, for a normal cell, you expect that there is a distribution of cargo, some are towards here, some are towards this end, and so on. Now, if somehow you manage to deactivate one sort of motor, so for example, if I deactivate my dynein motors, then what I would expect from this simple mechanical tug-of-war sort of picture is that the kinesins would then take all your cargo towards the plus end. So if I were to look at the cells, I would see clusters of cargo towards the plus end, so towards the tips of these. On the other hand, if I were to deplete kinesin, then what I would expect is that the dyneins would then have no opposing motor, and they would simply walk towards the cell body or towards the shaft end of it. And then I would see clusters in this end. So this is a cartoon of the expectation that I have based on the simple tug-of-war sort of a thinking. What the experiments see is different. What the experiments see is that, so these are pictures from the control cell. This is where you have depleted the dynein. So your mutation in the dynein heavy chain. This is where you deplete the kinesin heavy chain. And what you see is that nowhere do you observe such clustering either towards the plus end or towards the minus end. So in this case, dyneins are inactive, but still you have motors, still you have cargo present throughout the cell body. In this case, kinesins are inactive, but again you have motors, cargo present throughout the cell body. And in fact, you can do a quantitative analysis. So this yellow part over here are cargo which are in the tip region. The blue parts are where the cargo is in the shaft region. This is my control cell. This is where dynein is depleted. This is where kinesin is depleted. And I've forgotten what this is. That is kinesin 2. Okay. And you see that nowhere is that it's a purely yellow curve or it's a purely blue curve. So again, there is a distribution of cargo. So, and what they hypothesize in this paper is that what happens when you either deplete dynein or you deplete kinesin is that your cargo actually becomes immobile, and then they're carried towards the plus end just purely due to the polymerization of the underlying microtubule tracks. Okay. So here is one evidence. Where in spite of the fact that you have these opposing teams of, yes? So, sorry, if, So you're saying whether there is a remnant fraction of motors which is sort of enabling, I don't think so. I would have to go back to the experiments and check, but my understanding is that that is not so. In fact, if you sort of replace dyneins by a different -n directed motor, you again restore your transport. So here, contrary to a sort of naive expectation that if I sort of stop the activity of one sort of motor, you get transport in the other direction. Here you see that it's a very interlinked system. So this is called codependent transport, where if you affect one type of motor, you can actually affect transport in the other direction. These are again, I think, Drosophila neurons. This is the control cell, and this is cells where there is a mutation to the glued component of the dynactin, which is sort of a cofactor for dynein and helps in dynein binding and unbinding. And again, what you see is that what you get are clumps of cargo. So the cargo has sort of stopped moving, and you get these stationary clumps or traffic jams, what have you, which are non-motile. So there's this nice sort of review by Hancock, which talks about this bidirectional cargo transport and moving beyond tug-of-war models. So what are the problems of the tug-of-war models and how one can sort of move beyond? And this is one of the aspects is this, and this is what he calls the paradox of codependence, that even though we know there's plenty of evidence from existing experiments that tug-of-wars do happen, but in spite of that, you see this sort of crucial interdependence of one motor on the other, where sort of stopping one stops the other. Okay. So having said that, let us now focus on how dyneins are different from kinesins. There are multiple ways, and so I'll focus on one, which is this Dynein Catch Bond. So these are experiments from Professor Gross's lab and Ambarish in 2001. So what you do is that you have cargoes that are moving along your microtubule. Then at some point, so at t equal to 0, you switch on an optical trap such that you apply a force that is greater than the stall force. So the motors stay bound for some time before they detach. If you plot the histograms of the time it stays before it detaches, you see there is a sort of exponential distribution. So these are data for kinesins at different forces. These are dyneins at different forces. So you plot the histograms as a function of the detachment time. You see there is typically an exponential distribution. You find out the characteristic time of that exponential distribution, and then you plot the characteristic detachment time as a function of force. The inverse of this detachment time gives you the dissociation rate. How much does the dynein, at what rate does the dynein unbind as a function of the opposing load that you supply. So what you see over here is that kinesins have a sort of increasing nature. I'll sort of gloss over this kink over in this part, and I'll just say that this is an increasing curve. So as you increase the force, the dissociation rate of kinesin increases. This is the standard Kramer's escape theory. So you have a barrier to escape, and then this force sort of lowers the barrier to escape faster. On the other hand, dynein show this extremely counterintuitive behavior, where you're pulling on it with an opposing force, but there is a region where the larger the force you pull it with, the smaller the dyneins dissociate. So I'm pulling on it, but the dyneins are sort of catching hold of the microtubule more strongly. So it's harder to break the bond, which means the dissociation rate decreases. So this is a standard, this is a classic sort of catch bond rate for Dynein. And it has been seen in other experiments as well subsequently. So I think these are experiments from George Schubater's lab, where again you do similar experiments. You measure the residence time that the cargo, the motors stay attached before detachment. And then you can plot the residence time as a function of the opposing load that you're applying through an optical trap or whatever. And you see that this is for kinesin-driven motion, this is for Dynein-driven motion. And for kinesin-driven motion, as you increase the load, the residence time decreases, so the motors detach faster. For Dynein-driven motion, if you increase the load, the residence time increases. So this is again a signature of catch bond. These are experiments from Roop Mallik's lab, where again you plot the time of superstall as a function of stall force, which is like a proxy for the number of dyneins that are attached and shows the increasing behavior. So experiments have seen catch bond in this dynein as opposed to kinesin, where you always see a slip bond. So you always have the dissociation rate that is increasing as a function of time. So what we want to ask is that if I now want to incorporate the effects of this Dynein catch bond and try to predict what are the transport properties of this cargo that are carried by a slip-bonded kinesin and a catch-bonded dynein at the same time, how do I go about doing that? So to do that, you need to sort of have a mechanism to deal with these catch bonds. And there is such an existing mechanism. So catch bonds are known in various biological contexts. It's fairly recent for dynein, but in other contexts, it has been known for a long time. So this is a lifetime of a receptor-ligand bond under force. And again, you see that it first increases and then decreases. These are lifetimes of the actomyosin complex, where it's well known that, again, they have a catch bond. And again, you see this first, this non-monotonic behavior, where you have first an increase in the lifetime, and then you have a decrease in the lifetime. So how does one go about thinking about this? There are various pictures, mental pictures that one has when one talks about such catch bonds. So I'll talk of one of them, which is what we use for this paper. It does not really matter, the equations sort of give you the same result at the end. So this is what is called the deformation pathway picture of a catch bond. So the basic idea is that in the absence of a force, your bond is sitting somewhere in your free energy landscape that is not really at the minimum of the landscape. So if you now apply a force, you induce some sort of maybe conformational change that, sorry, that's, oh, okay, sorry. So if you now pull on it with some force, you induce maybe a conformational change that makes it go more towards the center of it, more towards the minima of your free energy landscape, and that stabilizes the bond, and therefore it makes it more difficult to break. It's a very sort of hand-waving picture, but it can turn out to be quite powerful. So in terms of a sort of barrier escape problem, so here is my black curve, which is my standard barrier. So my bond, my dynein microtubule bond must escape over this barrier in order to break the bond. In general, if you have a standard slip bond, what happens is you apply a force is that this barrier sort of goes down, and therefore it becomes easier to escape as a function of force, and that leads to this exponential sort of a behavior. So here what the claim is in this sort of a picture that we have, this deformation pathway picture that we have, is that it's true that this barrier comes down with force, but additionally, the minima of your free energy landscape, that also goes down with force. Okay? So you have two things that are going down. The minima is going down as well as the barrier is going down. So you have one, if I write down the dissociation rates, the unbinding rates, you have one contribution, which is my standard sort of Kramer's contribution, e to the power of f by some standard dissociation force scale. But additionally, in this case, because your minima is also getting lowered as a function of force, you have this dissociation force, Ed, which is a function of f itself. And you can have this sort of picture can reproduce both slip as well as catch, depending on how fast this barrier goes down as compared to how fast this minima of your free energy landscape goes down. You can reproduce slip-like behavior, you can reproduce catch-like behavior, and so on. Okay, so now you need something, you need some sort of expression for how this dissociation or deformation energy sort of behaves as a function of force. And one can write down a sort of very phenomenological force expression. What this says is that at zero force, you have your deformation energy is zero. As you go to extremely large forces, it saturates at some value, which is given by alpha. So this alpha over here is a parameter that parameterizes my catch bond strength. This F naught is a force scale that characterizes the response of the catch bond to force. I want to stress that this is a purely sort of phenomenological model. It's not like people have studied Dynein and said that from the microscopic structure and so on, said that this must be the deformation force form. But what it does is that it does manage to reproduce a range of processes very generally, this sort of a form. So if you look at these solid curves over here, this is for the receptor-ligand, this is for the actomyosin, this sort of a form for your deformation energy and then therefore the dissociation rate manages to capture the bond lifetimes quite well, even without going into the detailed biology of how this sort of an expression comes about. That's ideally what one would like, of course, to argue from microscopic details that this or whatever else is the form of the deformation energy, but we just for now proceed with this sort of a phenomenological form. Okay, so it's not very clear as to what happens in Dynein that sets this catch bond, at least as far as I know. There have been a couple of proposals. This is from Ron Vale's lab, and what they say is that this stalk region of your microtubule binding domain that has a tension sensing mechanism. So when you are under strong external forces, this sort of stalk goes to an angle which is greater than 90 degrees, and maybe then that has a strong affinity to this microtubule, to these microtubulin dimers, and therefore that's what sets in the catch bond. And this is a similar sort of cartoon. This is due to Roop and Ish, where this proposes a similar mechanism that as the stalk sort of stiffens under tension, you have this sort of receptor-ligand binding. So this is the end of the microtubule binding domain. This is part of the tubulin dimer where it binds to. And as the stalk stiffens, maybe there is a conformational change where this bond becomes more stable, and therefore it costs you more energy to sort of break this bond. But I don't know, as far as I know, I do not think there is any clear-cut understanding of why this catch bonding happens, or what is the exact mechanism. So in the absence of that, we sort of go along with this phenomenological deformation expression, except now that for Dynein, the deformation energy sets in only for forces that are greater than the stalled force, right? Because that's what's seen in experiments. If you have forces that are greater than the stalled force, then that's where your deformation energy sets in. Your alpha is the strength of the catch bond, F naught sets the scale of the force response. Okay. So if I have written down the deformation energy of this form, and therefore associated with it, I have a dissociation rate, I can go and ask, well, does that fit the experimental data that we have? And it turns out that it does fairly well, not exactly, but quite well, I would say. So so my alpha and F naught are free parameters. I don't really have any molecular understanding of what they come from. So what I do is that I fit the experimental data with this sort of a function, and then I can extract parameters for alpha. I can extract parameters for F. There's nothing really sacrosanct about these numbers. I will still play with these numbers a little bit as we go on and see what the effects are of different catch bond strengths and so on. So this is the unbinding rate. You can ask, so a couple of results I'll show for unidirectional transport. So where you just have teams of dyneins, no kinesins, and then I'll move on to the bidirectional transport. So if you ask what are the average number of bound motors as a function of force, this non-monotonicity gets reflected in the average number of bound motors as well. So as a function of force, the average number of bound motors first increases, and then as the catch bond sets in beyond stall, you can have an increase in the average number and then finally a decrease. This is the force velocity curve, which shows a monotonic decrease, which is fine. But where I wanted to show this, so if I go to the next slide, what you'll see is that if you plot the probability for a bound motor state, for a state which has no motors bound, or one motor bound, or two motors bound, and so on, for two cases, one is where you do not have any opposing force, and the second is where you do have an opposing force. What you'll see is that what this catch bond does is that it stabilizes states with these higher number of bound motors. So these green bars are the case of zero force. These red bars are the case where you have some 9 piconewtons of force. And you'll see that compared to the green bars, these higher motor states, so 2, 3, and so on, these have higher probability compared to the zero force states. And that can, in fact, get reflected in the force velocity curves in certain parameter regimes. So I think these are some These two curves are two attachment rates for dyneins. This is about 0.1 per second. This is about 1 per second. So the parameters are roughly in the biological regime. We haven't chosen anything that is completely off. And what you can see is that the fact that these higher motor states get stabilized can show up as these non-monotonicities in the force velocity curves as well. It's an open question. I do not really know whether one can resolve humps of this sort from an experiment, but if possible, then this could provide one testable prediction theory. And then one can characterize phase diagrams as a function of stall force. So Dynein has two sorts of stall forces. In mammalian dyneins, your Dynein stall force is around 1.1 piconewton, whereas yeast Dynein has something around 5 to 7 piconewtons. So there is a range of stall forces that you can have. So what we just wanted to ask is that if I plot a phase diagram of this deformation strength, my catch bond strength, alpha, as a function of this stall force for two different values of the attachment rate, the pi attachment, then how many humps do I see in this force velocity curve? This violet corresponds to where you have a monotonic decrease, and then red, blue, green, blue, yellow correspond to where you have 1, 2, 3, 4 sort of peaks. Okay. This is something we published last year. But let me move on to the main part, which is this case when you have bidirectional transport. So you have, we go back to this picture, where you have teams of dyneins as well as teams of kinesins. The formalism is exactly the same as the Mueller-Lipowski paper. So you write down the master equation, depending on the number of attached dyneins and the number of attached dyneins, which is n minus, and the number of attached kinesins, which are n plus. You have some expressions for how your binding rates go, and these are independent of force. You have some sort of a velocity model. And then for the detachment kinetics, for kinesins, we use the standard slip bond. And for dyneins, we use this model that I have just described, which can experimentally reproduce the single dynein behavior. So you then stochastically simulate these equations. You look at trajectories, and then you ask, how do the transport characteristics change? Okay. So I'll just I'll show some results. Oh, I forgot to mention. So this is a situation where you have some maximum number of motors that can bind. So you have some capital N plus number of kinesins, some capital N minus number of kinesins which are bound to the cargo. Not all of them are bound to the microtubule. The number which are bound to the microtubule are the small n's, but these small n's can vary between 0 to capital N. So there is a maximum. So this is a plot where the maximum number of kinesins that you can have is 4. And what I'm plotting is the average run length. So the average run length is the average length the cargo walks before it detaches. And my detachment is defined as the point when it reaches a 0-0 state. So when you have no motors bound, then that's where it detaches. And that's what I calculate as my run length. I do this many times, and I get some sort of an average run length. So what I'm varying over here is the number of dyneins, the maximum number of dyneins that I can have. So if I first look at the non-catch bonded behavior, which is this green and the black, so I've plotted for two FSs, 1 and 7. What you see is that as I increase my number of dyneins, my average run length sort of in the positive direction sort of decreases, which is fine, which is what I would expect from a standard war sort of scenario. I'm having more and more motors, more and more minus directed motors. What that does is that sort of reduces your run length in the plus direction. On the other hand, if you now have catch bonded dynein, so let's first see this curve, which is Fs7 piconewton and some catch bond strength, which is like 40 kBt. What you see is that in contrast to this sort of smooth decrease in the non-catch bonded case, even 1 or 2 attached dyneins is enough to sort of bring down your run length to almost 0. So a catch bonded dynein can actually, even a single or about 2 catch bonded dyneins can actually stop strong positive directed motion for 4 kinesins, for example. The effect is even more stark if you consider the dynein with a stall force of 1 piconewton. And here it again stalls for about 1 or 2 dyneins. And then if you increase the dynein number, it starts walking in the negative direction. In contrast to this case where you did not have any catch bond, where you decrease your run length, but you were still always walking in the positive direction. Here, as you increase your N minus, you can have highly precessive runs in the negative direction. And this is a case where you always stall the cargo as you increase the number of dyneins. So this range of behavior that is possible, and you do not really need to match the dyneins and the kinesin numbers in order to see this sort of an effect. This comes purely because of the nonlinearities that are manifested in the catch bond. So this is purely a catch bond effect in the average one. This is a similar curve, but this is, so the earlier one was a case where you had a maximum of 4 kinesins. I'll just show one more where you have a case where you had 9 kinesins. So again, it's extremely strongly, walks very strongly in the positive direction in the absence of any catch bond. Now, if you have a catch bonded dynein with a 7 piconewton stall, what you do is that you still see this sort of monotonic decrease, smooth continuous monotonic decrease, but the amount of decrease is much larger as compared to non-catch bonded dynein. On the other hand, if you have this 1 piconewton dynein, the weak dynein, again, now maybe 1 is not enough, but around 3 to 4 dyneins are enough to counteract 9 kinesins and bring the motor to a stall. The reason why this one is more is effective is because in this case, the catch bond sets in much earlier. It sets in at a force of 1 piconewton as opposed to this case where the catch bond sets in at 7 piconewtons. So you see the catch bond effects more strongly for this, this class of dyneins, so like mammalian dyneins putatively, rather than the 7 piconewton dyneins. Okay. And there is some sort of evidence that you do have only 1 or 2 dyneins that are counteracting teams of 4 or 5 kinesins. So these are experiments from 2011. Forgot what this is from. But they do a quantitative photobleaching sort of experiment. And so where you have both dyneins and kinesins attached simultaneously in the cargo. And what you see is that the dynein has a very strong peak at around 1. There is a much smaller peak, a much weaker peak at around 2 dyneins. Whereas if you look at the kinesins, I think I took an intermediate curve, but whatever. If you look at the number of kinesins, you'll see that the probability distribution has a peak at around 4, 5, 6, that sort of a number. So you can have a very few number of dyneins that are counteracting larger teams of kinesin. You can now do the opposite thing. So earlier what we did was that we varied the number of dyneins. So these would be experiments where you sort of inactivate dyneins and that would change the dynein number, the active dynein number. You can do the other thing where you fix a number of dyneins and then you change the number of kinesins and ask the same thing, that how does the average run length behave. So this is, I have 4 dyneins and then I will change my number of kinesins and ask what happens. So in the non-catch bonded case, so in this case, it starts off very small and then rapidly beyond about 4 kinesins, it starts walking very strongly in the plus direction. Now if you have catch bonded dynein with the 7 piconewton stalls, you see that this region where you have very small run lengths that continues for a longer time, so it's till about 6 kinesins, and then it starts to increase. But strikingly what happens is, if you consider this 1 piconewton dynein, where what you're doing is that you're increasing the number of kinesins, but what your cargo is doing is that it's moving more and more in the negative direction. Right? Because these kinesins, what they're doing is that they're exerting a force on the dyneins, which is sort of enough to make it go into this catch bonded regime. Once it goes into the catch regime, it sort of holds on to the cargo much stronger, and then it walks more processibly in the negative direction. Eventually, of course, if you increase your number of kinesins large enough, you'll see a reversal of this characteristic, and your run length will sort of decrease in the negative direction and finally go positive if you go to large enough numbers of kinesins. You can see similar behavior. For, so this is the case where I have 9 dyneins, and then again I vary the number of kinesins, and again you see this strong sort of counterintuitive behavior when I'm increasing the number of kinesins, and they send the dynein into a catch regime where dyneins now walk more towards the minus end. So you have these signatures of this sort of codependent transport where affecting the properties of one class of motors instead of pushing motion in the opposite direction can have these various sorts of counterintuitive properties in the run lengths and so on. And just to convince you that this is these numbers that I have chosen, They do not mean anything special. These are just some representative numbers that I chose. We have contour plots of this run length in this n plus n minus plane, both for non-catch bonded as well as for catch bonded dynein. So this is cut off. This is FS1 piconewton. This is 7 piconewton. And this region over here is where you see this sort of a reversal. So this blue region over here is where you see that reversal, where you're increasing the number of, even as you increase the number of kinesins, your cargo walks more towards the minus end. So this is a robust feature in certain regions of the phase space. For this non-catch bonded thing, the moment you have maybe beyond 4 or 5 kinesins, you sort of walk very processively towards the plus end. That's not the case here. You need very high number of kinesins, very low number of dyneins to walk large distances in the plus region, whereas here you have this very strong counterintuitive phenomenon of the negative directed runs increasing. Okay. So what I did was earlier I changed the number of kinesins and dyneins, but you can weaken your motors in other ways. So for example, you can weaken the unbinding, zero force unbinding rate as well. That might correspond to these experiments where you play around with Dynein, which will affect the binding and unbinding rates. So these are average run lengths for a particular set of n plus n minus. So I just chose 6 and 2 to stay close to sort of experimental numbers. My attachment rate is 1 per second, and then I vary my detachment, my zero force detachment rate, and see what sort of behavior I have for different stall forces of dynein. You can see that, so for example, for 1 piconewton dynein, as I increase my attachment rate, so as my dynein becomes weaker, increasing epsilon naught means dyneins unbind faster. So as they become weaker, my run length in the negative direction decreases, as I would expect. But then beyond a point, it does not change anymore. So it sort of stays stationary. At 2 piconewtons, again, it decreases at first, but then you have this sort of counterintuitive behavior again, where I'm weakening my dyneins, but my run length in the plus direction also decreases. And you can see this very robustly for this 4 piconewton dyneins, where you have a consistent decrease as I continuously vary epsilon naught minus. Finally, this is the normal this is the normal case, the one that I would expect, that as I weaken my dyneins, my run length in the positive direction increases. So you can have all these different sets of behaviors from the same theory, depending on where in parameter space that you are. And again, we have contour plots for these. So this is the run length contour plot in this stall force detachment rate plane. And you see that this is the region where I had this sort of a curve. And this region over here is where I had this red curve. This green curve is somewhere over here, around 4 piconewtons, and the normal behavior is over here. And again, I really do not have too much of a handle on, or at least biologically, I do not really understand what this catch bond strength is. So depending on what you set your alpha to be, you can get different sorts of phase behaviors of this average run length. Okay. So one final thing that I want to say is that often what we do is that we look at motility diagrams. And in motility diagrams, what you do is that you take the master equation that I write down, you look at the steady state, so you set your del P del T's to 0, and then you look at the steady state probability distribution. You can then characterize your motion in terms of the peaks of the steady state probability distribution, and that's what gives you your motility diagram. So for example, if you have n plus much greater than n minus, then you would have maybe strong plus directed runs, so which I would call, or this is from Lipopsky's paper, which would be called plus. If you have no motion, so the cargo spends most of the time in the pause state, then that's a 0 and so on. So you can construct the corresponding motility diagrams for these cases with the catch bond, and they do not really bring out this feature of this non-monotonicities in the run lengths. So this run length contour diagram actually does not have too much of a correlation with the motility diagrams that one can calculate from the steady state probability distributions. So in this case, for example, the motility diagram looks fairly featureless, whereas the run length contour diagram looks a lot more, it has a lot richer features which are actually evident in the plots that I just showed. On the other end here, there is a very complicated motility diagram, which does not really find a signature in this Runlength contour plots. Okay, so this is more or less it. So what I wanted to say is that if you have a tug-of-war model, a purely mechanical model still, but which incorporates this Dynein Catch Bond characteristics, the experimentally determined characteristics, then you can reproduce both conventional tug-of-war as well as this codependent motion. So you can have both the fact that if I decrease one, the other increases, or the case where it does not. This is by no means a sort of complete theory. The first thing that one should do is to relax this assumption of equal load sharing, and this is something that we are doing now. What happens if the load is shared stochastically between motors? Then there is this whole story about ATP binding and ATP hydrolysis and how that couples to the step. So there is this kinesin and dynein mechanochemistry. There is another very interesting feature of dynein, which I did not talk about, which is that dyneins have variable step sizes. So if dyneins are not under any opposing forces, they walk with step lengths of around 32, 24 to 32. Then as you pull on them more and more, they adapt so that they reduce their step sizes. So it can go from 32 to 24 to 16 to 8 nanometers. This is in opposition, this is in contrast to kinesin, which always walks with 8 nanometer steps. So you have this variable step sizes for dynein, which can also play a role in how it adapts to opposing forces. And finally, there's this thing that Stefan talked about, which is regulation due to various cofactors, which would also play an extremely important role, especially in vivo transport. So hopefully we'll at least get some of these done. Thanks. Okay, so this is work done with Abhishek Chaudhary at Aisan Mohali and Shudipta Mohuri at Pune University. Most of these stochastic simulations were done by Palka, who was an undergraduate student in IIT and is now working with Stefan at Göttingen. Shupriyo was a master's student who set up this initial motility calculations, and he's now at ISC, Prabal. And Anil and Sameep worked on this first paper for directional transport. And funding is from DST. Thank you. Yes.
I guess I'm a little confused by this plot or these plots where you held the number of, let's say, dyneins fixed and varied the number of kinesins. Yeah, this kind of thing here. Yeah. And then you basically get motion in the minus direction. So the dyneins, when you exceed the stall force, then they begin moving again. Is that what this implies?
No. So what this implies is that yes. So you have a backward load on this dynein, which is greater than the stall force.
Right.
So what you're doing is that these dyneins are sort of attaching to the microtubule more strongly.
Right.
Okay. And then it starts moving again when it goes beyond stall force. So what it does is that it reduces the detachment rates of dynein so that you do not go to the zero dynein state as easily. So it holds on for longer, and that reflects in the run length, because I'm calculating the run length until I reach the 0, 0 state.
Okay. I'll have to think.
I can do.
Any other question?
Sorry. What is the advantage of?
Yeah, I mean, rather than having, like a very linear, intuitive method, like it is a counterintuitive control of the transport, right? I mean, how does it benefit this cell transportation?
Okay, so there are two aspects. One is that this is what is seen in experiments, that these are linked very closely. It's not a naive thing that you switch off one and the other increases. So that helps. I cannot give you a precise answer. Maybe one of the Professor Gross or somebody can. But it sort of helps the cell exert a more precise control of how long the cargo travels and in which direction. So by just changing the motor numbers a little bit in this way or that way, it can get directed motion towards one end or the other. Maybe.
So I have some doubts about whether or not, because basically what you're saying is that with the catch bonds, you induce lots of tug-of-wars, right?
Yes.
And then the dyneins lose in the tug-of-wars, and that sort of accounts for this. I mean, what we know from experiments is that the feedback seems to be a lot more subtle, that when you do, say, a knockdown of half of the motors in one direction, there's inactivation of half in the other direction. So, for instance, the cargos still move just fine. It's just that forces, for instance, indicate that you've downregulated a subset of your motors to balance. So it doesn't seem like it's a big tug-of-war type thing that inactivates everything. But that isn't to say that this couldn't happen. Just that I have some doubts about whether it's the sort of predominant way that things are regulated.
I guess probably the thing is that, what we were talking about that day, that it would have to be a coupling of this sort of a mechanism with the regulation that would...
Yeah, I think that there's a heavy aspect of regulation in here.
But what I think, so we want to build on the complexity, but what I just wanted to say is that this would be one of the mechanisms that would play a role, and possibly. And
yes, if you have tug-of-war, so I think there's a lot of debate about exactly how many tug-of-wars you really have. In some systems like the endosomes, there does seem to be some evidence from Roop's extension. But in other cases of bidirectionally moving cargos, there's much, much less evidence. And it seems that, like in lipid droplets where we've studied it, mostly there's coordinated regulation. So for instance, when you stall a cargo and it falls off the microtubule and then rebinds, as was pointed out in like Stefan's, there's a memory effect. And often in our case, when it falls off and rebinds, the time that you have to wait before it rebinds is quite long, suggesting, like 10 seconds or something. And that has to reflect regulation, because if you assume that there are 5 or 6 motors and they were all active, you would get binding within, less than a second. And if you assume that the on rates are so slow that it takes them that long, then you would never get 5 or 6 motors working together. So I think you have to, at least in a lot of experiments, you have to invoke some aspect of regulation, which doesn't mean this kind of thing doesn't happen in addition. But